Okay, so today in the third lecture, we will get into, using mutants to identify genes. Okay, so when I say mutant, it means that it has properties different from the normal one, which we call the wild type. As I said, that normal can be subjective. Okay, here we take something as normal, which is internationally recognized. One particular, genotype having a particular set of phenotypes, that's called normal. So that is wild type. Okay? Now, if you think from a diploid organism's point of view, then if both the copies in the genome are wild type, then the phenotype will be wild type. If one copy is mutant, and that copy happens to be a dominant phenotype, or that copy happens to be the dominant allele, then the phenotype will be mutant. Now, I want to be very, very categorical here that a mutation does not necessarily mean loss of function. Gain of function can also be a mutation. As I gave an example in the last class, that you have a mouse that has twice the size of the brain of a normal mouse. Is it bad? Good? I don't know. But it's a mutant phenotype. It's not normal. Now, if the change in genetic situation is such that the genetic sequence is such that the altered version, the other allele, is dominant over the normal allele, then the phenotype that will be seen is of the dominant allele. So even if it is heterozygous, it will still be mutant. And of course, homozygous will be mutant. But that is not the case in the case of recessive. In a way, you can think that in the case of a recessive mutation, the wild type allele is dominant, because even though it's heterozygous and the other copy is a mutant, even if it is a loss or gain of function mutant, the phenotype will still be wild type. Okay? So a recessive mutation in the heterozygous that does not display any phenotype. They look, feel like wild type. However, if it is homozygous recessive, then the phenotype will be that of the mutant. Okay. Again, coming back to the meiosis, you look at it like this, that the way the genotype is denoted is that the capital A in yellow is the mutant genotype, and small a in blue is the wild type genotype. Okay, it's kind of just to show that it's the dominant one, it is the capital one. Okay? Now that is in the diploid organism, it is capital A over capital A in the mutant, and in the wild type it is small a over small a. The gametes, meaning the haplocontent cell, which is gametes, in yeast, which is spores, they will have only one copy of the gene. Now, in this particular case, it has no option. Okay, if it is coming from the yellow parent, then no matter what it is, the only copy will be the capital A dominant mutant, and the other copy will be in the other gamete, it will be the small a, the wild type one. Now, if these two gametes fuse to make a new offspring, the first filial generation of F1, or like in our case, it is the zygote, then it is getting one copy of the gene from the yellow gamete and one copy of the gene from the blue gamete. So it will be capital A over small a. But because capital A is a dominant allele, so its phenotype will be seen through. Therefore, the heterozygote, capital A over small a, will have the mutant phenotype. Now, this F1 generation heterozygote, it is heterozygote, right? Because the gene is, the allele is not the same in both the copies. It's a heterozygote. Now, when you think of the next generation, F2 generation, this heterozygote will produce gametes of two kind, capital A or small a. Now, if the male gamete is capital A and the female gamete is also capital A, then the F2 generation diploid will be capital A over small a, that is mutant. If the male gamete is capital A, but the female gamete is small a, then the F2 generation will be a heterozygous, capital A over small a, but its phenotype will still be mutant. If the male gamete is small a, but the female gamete is capital A, then the F2 generation, small a over capital A, will be a mutant. Because in all these cases, at least one copy of the gene is capital A and that is dominant. Therefore, the mutant phenotype will be seen through. Only in 1 in 4 probability, it will be small a over small a in the F2 generation, and that will be wild type. So if it is a monogenic trait that we are talking about, like only one gene, A, one locus. Then if that trait, if that allele is dominant, then in the F2 generation, 3:1 will be the ratio of mutant is to wild type. However, if it is a recessive allele, that is, the mutant is shown as capital B over small b, and the wild type is capital B over capital B. I repeat, the mutant is small b over small b, and the wild type is capital B over capital B. Then you do the same thing, and now you remember that unless both the alleles in the diploid is small b over small b, it will behave as normal, okay? Because small b, the mutant, is a recessive allele. So if you made a recessive dominant, the homozygous recessive mutation with homozygous wild type, then in the F2 generation, normal is to mutant ratio will be 3:1. Okay, so that is actually this kind of a cross of a mutant with a wild type and observing the ratio of normal versus mutant is a good test to examine if a particular allele is a dominant allele or a mutant allele. There are two assumptions and critical concept issues that I would like to clarify here. Number one, the assumption is that in a particular organism, cell, gamete, whatever you call, there is one and only one mutation Everything else is just like the wild type. That's a critical assumption, and experimentally is correct that you can work with that kind of an assumption. I will elaborate when I come to that part of the lecture. The other assumption, it is not an assumption, it's a mathematical fact that it is a probability. Okay? So if you have 8 progeny in F2, all of them are mutant, you cannot automatically say that the mutant was the allele was a dominant allele. It's just a matter of chance. So we need to increase the number, like how many, etc. That's a question of statistical analysis. I'm really not good at it. Mathematics and I don't gel well. But typically, like you're talking about few thousands, and then you look at the ratio, it will come close to the ideal ratio. If in that kind of a situation, it is 1:3 mutant to wild type, you call it recessive allele. If it is 3:1 mutant is to normal, then you call it a dominant allele. Okay? So that's just a way of looking at it. Now, few things we will now get into yeast genetics. So yeast has an advantage which you and I don't have. Yeast can exist as haploid or as diploid. So if you challenge yeast cells with a nutrient limiting condition, particularly nitrogen limiting condition or starvation, they will undergo a process called sporulation, where a diploid yeast cell will undergo meiotic divisions and will form 4 spores. They are much more resilient, they're heat resistant, so they can survive for a very long time. This is adaptation to withstand hostile conditions and propagate its progeny. Okay? Now, geneticists take advantage of that because it's the same organism, same genome. You can just by manipulating the nutrient condition, you can make it either diploid or haploid. Okay? You can take 2 haploids. These haploids, they express a pheromone. These pheromones act through G protein coupled receptor signaling pathway. They change a whole lot of things in the recipient cells, and that induce mating. Now, if the cell produces the mating pheromone alpha, then it is called a type A, mating type A, MAT A yeast. If it produces the pheromone A, then it is called type alpha or MAT alpha yeast cell. Okay? Now, only when you take 2 haploids that are of opposing mating type, then they will form a diploid. So in this particular case, one haploid is a mutant for the gene allele B, and the other one in capital in blue, that is the wild type version of the gene B. So the diploid will be capital B over small b, just to rehearse, because small b is a recessive trait, therefore it will behave as wild type because the mutation is recessive, and then you sporulate it. Okay. When you sporulate it, meaning you put it in starvation condition, then from each diploid yeast strain, 4 spores will be produced. And these 4 remain packed together. Okay? They like a big ball, within that 4 small balls are there. So the small balls of the spores, and they're covered by a membrane. you break the membrane, collect the 4 spores from a particular diploid and line them up. In here, the way you want to look at this plate is as individual rows. And you see only 2 black dots. Okay, those are the places where the haploid yeast cell divided and grew. So it formed a colony. Now, please note that 4 spores were placed on each of the verticals, but only 2 because the mutation is such that, because see, like B is a recessive mutation. In a diploid, the wild type version of the B will make sure that it behaves normally. But if it is a haploid and the only copy of the gene is B and it is not functional, then depending on the particular condition, the haploid cell may not be able to grow. Let us take an example. Let's say that the gene in question, B, capital B or small b, the gene in question is responsible for growth of yeast cells in the presence of galactose. Okay. Now the wild type will have all the genes for galactose metabolism. It will grow happily. But the mutant, the yellow colored spores carrying the recessive mutation, for them the only copy of the enzyme that is necessary for metabolizing galactose is non-functional. Therefore, that particular haploid will not be able to grow. So of the 4 haploid spores that you get out of the diploid heterozygous, 2 in light blue, capital B, they will be able to grow and the 2 will not be able to grow. Now, this is this 2:2 segregation in a recessive mutant is a very good indication that the mutation is in one gene and one gene alone. Okay? If the mutation was in more than one genes, then all the spores could have been affected. But statistically, if you see in a very large number, it is the phenotype is always segregating 2:2, segregating meaning you get 2 normal, 2 mutant. Then you can tell that this is monogenic trait. Okay? The behavior is because of one gene, and that particular gene is involved in galactose metabolism somehow. Now, this is a case when you have the mutation already there. You may or may not have caused the mutation. You already have a mutant cell. So you cross it with the wild type and then you sporulate it. Like if you want to see in the yellow spore or the yellow haploid whether its mutant behavior is because of mutation in one gene or in multiple genes. You don't worry about the blue one, right? Because it is wild type. It is behaving as wild type, normal. So then you cross it with the wild type, and then the resultant diploid, you sporulate it and segregate the spores, and you see whether the trait or the character or the phenotype that is not being able to grow in the presence of galactose as the only source of carbon is segregating 2:2. If it segregates 2:2, you conclude that the defect in the original parental haploid yellow colored small b is because of mutation in only one gene. Okay? Now, I must tell you that soon you will learn that we can actually create mutation in yeast. Okay. Now, if you have any idea that the mutation may be lethal because the gene that you are trying to mutate is an essential gene, like say a gene involved in DNA synthesis or RNA synthesis, not the regulatory part, the actual synthesis part, RNA polymerase subunit or DNA polymerase subunit, or something like that, essential. If you mutate that gene, then the haploid itself will not be able to grow. And in any medium, it doesn't matter whether it is glucose, galactose, or any kind of medium that you may provide. If there is no possibility of DNA synthesis or RNA synthesis, simply that haploid will not be able to divide further and form a colony. Okay? So those are essential genes, and if you have any indication that the gene that you are trying to work with is essential in nature, then you do the mutation in the haploid. Sorry, in the diploid. You do the mutation in the diploid, and then through tetrad dissection, you see whether the spores are segregating 2:2, meaning 2 are surviving and 2 are not. Then you know that that your suspicion is correct. The mutation that you affected was actually in a essential gene. Okay? Just a side information, we will have to remember it. But for most other cases, you will do your mutation in the haploid. Why do you want to do your mutation in the haploid? Because chances are, and for most genes it is true, that the mutation will be a recessive mutation. If you even lose a gene, if it is a diploid, the other gene, wild type copy, is present, and that may itself be sufficient to conduct normal activities of the mutant cell. Therefore, even though you have caused mutation in an important gene, you will not be able to see that the mutation has happened because there will be no phenotype. You cannot, please try to understand that all the examples that I will give you, like in the next few slides, you cannot tell by looking at the cell whether the DNA sequence has changed or not. Your only handle is that if the behavior of the cell is changed or not. It may not be able to grow in a higher temperature anymore. It may not be able to grow in presence of galactose as a carbon source. It may not be able to grow in presence of caffeine as a caffeine chemical challenge. Any or many of these things. So until or unless you have a behavioral test, phenotype, it is very difficult for you to ascertain whether a particular haploid cell has undergone any mutation or not. And as I said, that as most mutations will be recessive in nature, so if you conduct the mutation in a diploid, then in all the cases where the mutation is recessive, meaning the gene that you are mutating is not haploinsufficient locus, you will not even know that the mutation was done and successful, because the wild type copy will mask the effect of mutation. Therefore, in most of the cases, you conduct your mutation in a haploid cell. Okay. Now, let's get into the first classical experiment that is used for connecting genotype to phenotype. Now, yeast cells, they normally at 37 degrees centigrade. They can normally grow at 37 degrees. Now, if a gene sequence is mutated, please do understand what I'm trying to say. It is not universally true. If the gene sequence is mutated or changed, in a subset of such changes, a protein will be produced, like typically in a missense mutation. A protein will be produced whose thermal stability will be less than the wild type protein. That's possible, right? Some particular fold, some hydrogen bonding is missing because you have replaced a serine with an alanine, okay? Or you changed an acidic amino acid to a basic amino acid, and therefore its electrostatic interaction with neighboring amino acids is lost, and one or many of these things, which reduces the stability of the protein somehow. If that happens, then the protein is more likely to lose its structure at an elevated temperature than at a slightly lower temperature. So the protein itself is thermolabile. Thermo is heat, labile is not stable. So you do not know which particular aspect of the cell you are trying to probe. And this is where your first set of genetic experiments in eukaryotes, and even true in bacteria. You do not exactly know which phenomenon, which gene type you are looking at. You just want to identify all the genes which, if mutated, the yeast cell cannot grow at an elevated temperature. So these are called temperature sensitive screen. Okay. Temperature sensitive screen. You are screening for yeast cells that will be sensitive to higher temperature. There is a cold sensitive screen as well, less popular. Now, you have no clue which gene you should mutate. So you have decided that I will mutate all genes. But you have to take one precaution, that you will mutate only one gene at a time in every yeast cell. So one yeast cell should not have more than one mutation. Now that is a purely probabilistic calculation. Even though you have tried, you will end up having some cells mutated more than once, but majority of the cells will not have undergone any mutation because you have titrated the mutagen at such a level that it is very low. It is just good enough to cause mutation in a few cells, and you want to ensure that it does not cause more than one mutation per cell. So you will use a low enough concentration that many of the cells will not end up getting mutated at all. Some will get mutated. Of the some that will get mutated, some will be in essential genes or haploinsufficient genes. If they are mutated, they will their function is lost. Those haploids will not even grow. So you will not even know that certain other mutations also happened, which happened on essential genes. But if you leave these two extremes aside, that because of your very conservative approach where you have titrated the mutagen, majority of the cells will not have any mutation. And the other extreme is that they will have mutation, they will be in essential genes. neither of these serve any purpose. If it is an essential gene mutation, then you will not even get to see them. You will not even know that those mutations happen because those are lost in your experiment. And those cells that will not have any mutation or any mutation that has any effect on the survival or growth of these cells, you will not know whether those cells are mutated or not. Okay? So leaving these two extremes aside, you take a mixed culture, grow it in presence of some kind of a mutagen. Most of these mutagens typically will incorporate in the DNA, and they will behave like a nucleic acid. And then during further round of replication, they will change the sequence. You just want to ensure that in one cell, one modified nucleotide, that is your mutagen, is incorporated only in one place in the whole genome of the cell. Okay? How it is done is, little complex. I will give you a couple of papers about the art of this genetic screening. Please read those review articles. If you do not understand, come back to me. I will try to explain. Now, you do that, and then what is your objective? Your simple objective is to assess whether the cell can grow in presence of at a higher temperature or not. Now, first you want to ensure that you do not kill because you will see why. So first you kill the cells. After mutation, you plate them on a petri dish containing agar and yeast growth medium. You grow all the cells at the permissive temperature, lower temperature, 23 degrees. And then you make two copies. The way it is done is, you take a piece of velvet cloth or a paper, nicely put it on the petri plate, and then lift it, go to the next petri plate where nothing is there, clean, freshly autoclaved, sterile petri plate. You very gently rub on the paper, and some parts of the yeast colony, haploid yeast colony, will be transferred to the new plate. And you can make two copies like this. And one copy of a particular plate, you grow it at the permissive temperature, 23 degrees centigrade, and the other one you grow at a higher temperature, 36 degrees. You will find a few cells that will grow at the 23 degrees centigrade permissive temperature, but not in the 36 degrees centigrade non-permissive temperature. So you now know that, okay, here is a mutant, a temperature sensitive mutant that can grow under permissive condition, but not under non-permissive condition. Then, it becomes your job, next 3 years, 4 years project, sometimes longer than that, to figure out which gene is mutated in here, why mutation in this particular gene causes stop of cell division, etc., etc. So this is how a temperature sensitive screen is conducted. Okay. So I will end here this particular lecture, and I will come back with lecture 4 where we start right from here. Okay. Thank you.